When we study humans, we're really studying a very sophisticated type of animal. That's right, animal. And you can tell all of your students that they're nothing but a bunch of animals. If they question you, you can always say, well, if you're not animals, then what are you, plants or protists? As we study the human beings, first we can put them in our classification scheme and see where they fall. Under the kingdom of animals, in the phylum Chordata, we find a class of mammals. One characteristic of mammals is that the young are nourished by the mammary glands of the mother, hence the name mammal. Next is the order of primates. And then to separate man from monkeys and apes, he's placed in the family Hominidae. There's only one species in this family alive on Earth today, and that, of course, is the Homo Sapiens. One of your main areas of study should be investigating the different body systems. One of the best ways to do this is to use one of these torsos. This particular one comes from Nystrom. If you don't have one in your school, check around your district. Check with a junior high or high school science teacher and see if you can borrow one for a week. We're going to divide this torso up into 5 major parts. First we have the head, and with a cutaway view we can see everything from the salivary glands to the brain. Half of the brain is removable, allowing us to see the different regions. The nervous system is made up of the brain and the spinal cord, which runs down inside the backbone. The second part we'll look at is what connects the head to the main torso body, and that's the neck. The third part are the limbs, and we can't obviously investigate them because this torso doesn't have limbs. But the 2 main parts that I'd like to spend some time on are these 2 cavities in the top and the bottom. This top one is called the thoracic cavity, and the bottom one is called the abdominal cavity. The thoracic cavity consists of the 2 lungs, which we see up here, and this lung is removable, allowing us to see the alveoli, or the tiny air sacs where oxygen is absorbed into the blood and carbon dioxide is deposited from the blood. Another system is the skeletal system, and it includes all the bones in our body, including the backbone. Here we see the collarbone and the ribs in a cutaway view. Now let's turn to the circulatory system, and the main organ of that system is our heart. This heart actually comes out, and I'll just remove half of it. You can see inside the heart that it's a 4-chambered heart. This is a characteristic of all mammals. Behind the heart, we have the trachea, which goes down to the lungs, and the esophagus, which goes down to the stomach. The abdominal cavity is separated from the thoracic cavity by a dome-shaped cover, which we call the diaphragm. When we start to look at the abdominal cavity, we learn a lot more about the digestive system. It's really quite amazing how our body takes in food and then automatically converts it into chemicals that our cells need to survive. To further understand this, it's good to review some of the topics that we already covered. First, we investigated the cell, and we realized that the cell needs certain chemicals not only to reproduce, but also to produce energy, to stay alive. When we did the experiment with osmosis, we saw that the starch inside the plastic bag wouldn't leave the bag because the molecules were too large, but the iodine would go inside. And when we looked at the elodea leaf experiment, we saw that when you add salt water, or water with a high concentration of minerals in it, it draws the water out from within the cell. It's important to keep these in mind as we study this digestive process. Let's start where it all begins, in the mouth. First, food is reduced to smaller pieces by the teeth. The tongue is important to help keep the food between the teeth. There are 6 salivary glands, 3 on each side. 4 of these are located under the mouth, and the other 2 are on either side of the face, near the ears. These are the largest salivary glands, and when they get infected with a virus, we get the disease called mumps. Now, saliva from these glands acts on food to produce a chemical change. It begins converting starch to sugar. Now remember that plant cells have a hard outer cell wall, and it's made of cellulose. Our body can't dissolve or digest this cellulose, so it passes through the body in the form of roughage. Whenever we eat starchy foods like potatoes, it's very important to cook them first so that we can burst this cell wall and the saliva can begin to act on the starch that's inside the cell wall. Now let's back up just a minute and look at what the cell needs. There's 2 main chemicals. Number 1, for the cell to reproduce, it has to have the building block of life, the protoplasm, the DNA molecule. And I don't want to scare you with a lot of big words, but DNA's main ingredient are what we call amino acids. And the amino acids are a derivative from proteins. So if we eat the protein, we need to convert it to an amino acid before the cell can use it. So one of the main functions of the digestive system is to make this conversion, turn the proteins into amino acids. The cell also needs sugar to burn with oxygen that we're breathing through our lungs, to give us the energy that we need, to give the muscles energy so that they can do work. So that's the number 2 ingredient. And it's the saliva in the mouth that begins to break down this starch into sugar. Now there's a good little trick that you can try on your students. Tell them to just start thinking about all the saliva building inside of their mouth, thinking about swallowing because their mouth is getting quite wet with saliva. Just thinking about all this saliva in your mouth really makes your mouth water, and it makes you want to swallow. How many of you have just swallowed? Well, let's go on now. The food has just been chewed up. It's been treated with saliva, and now it goes down through the esophagus. Think about the earthworm. There's 2 types of muscles, the circular muscles and the longitudinal muscles, and that's what makes the earthworm move. By squeezing down the circular muscles, it stretches its body out, and by shortening down the longitudinal muscles, it shortens its body down. This is what happens in our esophagus. It has the same types of muscles, and it pushes the food down into our stomach. This is how we can eat food while we're standing on our head and actually push the food up. Let's now take a look at some of these organs. First, underneath the liver, on the left side of our body, we find the stomach. The stomach is J-shaped and has muscles running in 3 different directions, which twist, squeeze, and churn the food. In the stomach, enzymes begin to break the proteins apart on their way to becoming amino acids. The liver is the largest organ in the body and chocolate brown in color. It has a number of functions. Bile is produced by the liver, which is stored in the gallbladder. Bile is an enzyme which breaks down fats into useful chemicals. It is added to the digested food after it leaves the stomach and enters the small intestine. Now there's another important gland located behind the stomach called the pancreas. It also adds enzymes to the small intestine, and the small intestine itself has glands which help to change the food. So we have bile, pancreatic fluid, and intestinal fluid, all of which react with food to produce the final products: glucose, which is a simple sugar, the amino acids, and the fatty acids. These materials are then transferred directly through the intestinal wall into the bloodstream. What is not transferred continues on into the large intestine. Here, water is absorbed and the mass becomes a more solid form of waste. Now we're involving the excretory system and the circulatory system, and it's really quite fascinating. The blood is a shipping company. It takes food and oxygen to the cells and takes the wastes away. And the liver, besides producing bile, is also a chemical factory and storehouse. It turns the sugar into a concentrated form and also stores, besides the sugar, important vitamins and minerals such as iron. Now when cells make new cells from amino acids, there are some waste generated. These are usually in the form of nitrogen compounds like ammonia. We must have an organ then to remove these wastes from our system. It's also important to regulate the concentration of dissolved minerals in the blood. Remember that Elodea leaf experiment. If the water's too salty on the outside of the cell, it draws all the water away from within the cell. The organ that handles this incredible task is called the kidneys. There's one kidney on either side of the body, behind the stomach and pancreas. The excess minerals and water that are collected are passed on via 2 long tubes to the bladder and eventually out of the system. The blood, now with just the right stuff, makes the long loop through the body again to pick up a new load. As students learn about the human body, they begin to appreciate all of these organs and the importance of not putting dangerous chemicals into the body that the organs have to deal with. And when you're teaching your unit on the human body, don't be afraid to use one of these torsos if you can get one. A good alternative would be one of those charts of the human body, or even have the students do a cut and paste activity where they're learning about the location and the function of the individual organs. Now throughout this series in Life Science, I've attempted to deal with the topic of reproduction in a low-key manner. I feel this is a good approach as students learn about reproduction from cells through plants and animals. Remember, it's the teacher who sets the classroom atmosphere, and the teacher who can have a major influence on how students perceive this topic. When you get to the subject of human reproduction, you should consult your principal or district office for appropriate guidelines. In California, for example, teachers must notify parents prior to any discussion on this topic, and then the parents have the opportunity to remove their child from this instruction. I believe that teaching human reproduction at a young age is very important, and I would make every effort to contrast it with the reproduction of other forms of life. Next, let's go on and explore other activities that help us learn more about the human body. A fun way to investigate the nervous system is to use a reaction timer. Here you can use yardsticks or pieces of molding from the lumber yard that are about 2 feet long. Let's see what I have here. First, I put a piece of tape on it. Then I lined up a small ruler with centimeters and marked off single centimeter increments. My scale runs from 0 to 40 centimeters, and I have a dark mark at the 0 line. Students work in groups of 2. One student is seated and the other one standing, holding the piece of wood. The student who's seated puts fingers on either side of the piece of wood. When the person standing lets go of the wood, the other person has to catch it as quickly as possible and then read the distance. Now we're talking about the time it takes from when the eye sees the wood falling to when the signal reaches the brain, and then the brain tells the fingers to do something about it. It's a measurable distance, and it can be converted easily to a time by using a table. I'll supply the table in the outline. Here's what I got when I did the experiment. First, my left hand seemed to do better than my right hand. The average of 5 trials on my left hand was 11 centimeters. The average of 5 trials on my right hand was 13. By using the table, I converted these distances into time and got 0.15 or 15 hundredths of a second for my left hand and 16 hundredths of a second for my right hand. You can also average these 2 times of both hands, which would be 12 centimeters, and then find the time for that. When you do this activity in your class, you can collect this last number from each student and graph the data on the board. Put the number of students in the vertical axis and the reaction time on the horizontal axis. Do a histogram if necessary. You should see a bell-shaped curve. Some students that are very quick, the majority of the students in one area, and then a few students that are a little slow. This also shows variations among the human race. By doing this activity, there's a number of advantages. Number one, we're learning how to do scientific experiments. We're also working with a metric system and seeing the relationship of centimeters to inches, particularly if there's a yardstick underneath this tape. Thirdly is that we're learning about mathematics and the importance of being able to find averages, for example, and also looking up information on tables. It's all valuable information. It all comes together in this one activity. When we continue investigating body systems, one thing we can look at is our eye. The eye is really pretty interesting. One thing we can do with it is make it move left, right, up, down, even go in clockwise or counterclockwise directions. What type of muscles must be involved to get the eye to do this? Well, if you have your model, you could show them the eye part of it and take a look at all 6 muscles that are connected to the eyeball. Remember that muscles contract and relax. When they contract, they get shorter. And when the top muscle contracts, it pulls the eye up. Combinations of muscles make the eye go in diagonal directions, and the 2 side muscles exert forces that are perpendicular to those of the other 4. When we move our eyes around in circles, we're using all 6 muscles. There are 2 types of receptors on the retina of the eye, the cones and the rods. The cones are used during the day. They help us to distinguish colors and to see sharp images. And the rods are used at night because they amplify the amount of available light. Some animals, like owls and bats, only have rods, and they can see very good at night. Other animals, like canaries and chickens, only have cones, and they're night blind. We can remember this distinction between the 2 by remembering night fishing. The rods are out at night. Now, where all of these receptors come together and form into a bundle and leave the eye and go to the brain, it's called the optic nerve. And that one little spot in the eye has no receptors. It's called our blind spot. We can visually identify that we have one with a little card like this. What I have here is a circle and an X on the card, and they're separated by about 10 centimeters or 4 inches. Here's how to use this card. Cover one eye and hold the card about an arm's length away and stare at the X. Move the card closer and closer until the circle just disappears. This is when the image is going through the eye and hitting that optic nerve in the eye where there are no receptors. If you move the card closer, the circle will reappear again. Now, if your students are working in groups of 2 or more, you can have them actually measure this distance when they can't see the circle and record it and see if it's consistent for all the members of the class. This is also a good experiment they can take home and try on their parents and make the same measurement. Come back the next day and see if adults have longer or shorter distances than they found for their classmates. Now let's go back to the muscular system. I'd forgotten about this one with the bathroom scale. Let the students squeeze the scale and see how much strength they have in their arms. See who has the higher value. You can always record the results here and get more involved in mathematics and graphing and that type of thing. When you're looking at the circulatory system, you can get a piece of clay and a long thin strip of wood like balsa wood. Form the clay into an oblong shape and flatten it slightly on the bottom. This is a pulse detector, and the longer the piece of wood, the more amplification you get. When you place it carefully at just the right spot on your wrist, the thin piece of wood will tick back and forth like a tiny metronome. Students can count the number of ticks in a 10-second time interval and multiply that number by 6 to determine the pulse rate per minute. Of course, they can measure the number of ticks in the whole minute interval too. But we're demonstrating the importance of mathematics, how we can make an operation more efficient by multiplying by 6. It's always a good question to ask your students why we chose that value 6 anyway. Incidentally, you can go on and do this experiment again after heavy exercise, record the results, and make comparisons of the pulse rate when they're resting and the pulse rate when they're breathing hard. Now to the skeletal system, and it's always interesting to take a look at x-rays. Check with your doctor or local hospital to see if you can get some x-rays for instructional purposes. Another thing you can do is check with your dentist. See if you can get some samples of teeth, maybe even start your own tooth collection where you look at different types of teeth from the mouth. You might even get a plaster cast of someone's teeth that you can let the students take a look at so they can see what the different types of teeth look like. You can also investigate bones. Go to your local butcher, get one of those large beef leg bones, and have the butcher slice the bone down the middle lengthwise for you. Take it back to class and let the students see what's inside of a bone. Some of the long bones are actually hollow inside and filled with a soft tissue called marrow. There are 2 types of marrow, red marrow and yellow marrow. The red marrow is usually found in flat bones like ribs, and the yellow marrow might be found along the inside of your leg bone. That yellow marrow is really fat, and students should be able to press on it and see how gooey it is. Now let's continue and look at 2 very interesting experiments that help us learn more about the human body. For this first experiment, I brought back our old friend bromothymol blue. That's the BTB solution that's in this container. Remember that when it's blue, it means it's a base, and when it turns yellow, it means it's an acid. When we bubble our breath through the blue liquid, it turns yellow. Now, since it's an acid when it's yellow, we go back to our chemistry ideas. We know that we can neutralize the acid with a base. So if it was yellow and we added a base to it, we could turn it back to blue. This is a principle we're working on in this experiment. What students will do is they'll start with exactly 100 milliliters of bromothymol blue in a cup. They'll blow their breath through it while at rest. In other words, they haven't exercised for 1 minute. After the minute's up, this will turn yellow. Then they'll take a dilute solution of ammonia, which is a base. And this is actually called a titration, but we don't need to scare them off with the big words. Add 1 drop of ammonia at a time into this bromothymol blue solution, stir it a little bit, add another drop, stir it a little bit, eventually it'll turn back to blue. The point that it turns back to blue, and they stop adding the drops and they write that number of drops down. Then they try the experiment again, but before they try it again in the active cup, they do some heavy exercise like 20 push-ups or jumping jacks or whatever, so they're breathing very hard. Bubble their breath through this one. And in fact, you're so out of breath you can't breathe through a straw, and they'll learn that right away. They need more oxygen in their body, and they're giving off more carbon dioxide. Now, even though the second one will look yellow, it still has more carbon dioxide in it. And when you do the titration with your ammonia here, you'll see that it takes more drops. Now, when I tried this, it took 10 drops from my at-rest one for a minute, and it took 14 to neutralize this one, to bring it back to blue. Each student in the group can use these same chemicals and try the experiment themselves. Remember that the number of drops is a measurement of the amount of carbon dioxide that's dissolved in the liquid. So when we're active, we produce more carbon dioxide, therefore we need more drops of ammonia to neutralize it. When you mix up this special mixture of dilute ammonia, you can really make it as dilute as you like. It'll just take more drops to neutralize it. This mixture right here is 10 milliliters of ammonia and 30 milliliters of water. It's a 1:3 ratio. And as with all experiments, you should test this one before you try it in your class. Nutrition is an important topic in the study of the human body. And while you're at it, why not do an experiment that measures the amount of vitamin C in different juices? First, we need our indicator solution, and it's easy to make. Take about 100 milliliters of water and add a pinch of cornstarch. Stir it up, and then you want to add iodine, 1 drop at a time, till it just turns the purpley blue solution like we're all familiar with. So I'll go ahead and do this. I'll try 1 drop and 2 drops, and then I'll stir it up again to see what kind of color we get. If it hasn't turned purple yet, you keep adding more drops until you get to the purple color. I'm adding 2 drops at a time. I'm cheating a little bit here, but it looks like it's starting to come up to the purple color that I want. We're going to be doing another titration, so the next measurement's very important. Each student will measure exactly 10 milliliters of this indicator solution and pour it in a test tube. We'll fill up 4 test tubes and set them in a test tube rack. Now this time we're titrating the indicator with the juice. Let's say in this test tube we want to test orange juice. See how much vitamin C is in the orange juice. Well, we get an eyedropper and we put some orange juice in it, and then we add 1 drop at a time and then stir it with a straw. If it turns clear, we know that there's a lot of vitamin C in it. If it still stays purple, we know that we need to add another drop. So we go to drop 2 and we do the same thing and stir it again. Eventually you get to a point where the vitamin C neutralizes the iodine indicator solution and turns it clear again. You record that number. The fewer drops, the stronger the vitamin C. If you used a mystery solution like water or something with no vitamin C in it, you just keep putting more drops and more drops in and it wouldn't change color. It would stay kind of a purpley blue color. So you can have all these stations around the room and let students go around and with each test tube titrate the indicator with the juice and see which juice is strongest. Now I tried orange juice, a vitamin C tablet that was crushed up and dissolved in water, and grapefruit juice from a freshly squeezed grapefruit. I found out that the vitamin C tablet had the greatest concentration. It only required 4 drops. The other 2 came in second at 6 drops apiece. You can try other juices too. You might even try those soft drinks that are supposedly fortified with vitamin C. Now this experiment is susceptible to contamination. It's important that students rinse out their eyedroppers when they go to the next station. And if they're using 1 or 2 test tubes, they should rinse those out thoroughly before pouring more blue liquid in them. And it's always good to have an extra test tube with some blue liquid in it for comparison, and put a piece of white paper behind it so you can compare the colors. Now, we've watched a lot of life science and you're probably pretty much experts, but there's one more topic we have yet to study, and that's ecosystems.